Thank you for the invitation to come and talk about some of the new data that has been generated with the new Kinnex kit and how we're using it in the diagnostics setting, particularly for rare diseases. And so the past couple of days, we've heard about rare diseases and how they are collectively actually quite common. And unfortunately, though, even with whole exome and whole genome sequencing, the diagnostic yield is still quite low. So it could be maybe slightly higher for certain disorders or within research, but clinically it still kind of remains at about 20 to 25%. And we know that about 80% of rare disorders have a genomic component, but only about 5% have approved treatments. So why are we missing some of these mutations and some of these diagnoses? And one of the many reasons is that we have this massive interpretation bottleneck, and this bottleneck is only getting bigger the more we sequence. We don't have the same ability to kind of interpret the data as the same amount that we are actually making it. And some of the reasons for this is that we've got a very protein-centric way of looking at variants. And what happens then is that we often end up overlooking variants that affect splicing or variants within the non-coding region. And this is because it's really hard to predict the impact of these variants just with the DNA alone. And I think a really great example of this is this preprint from Nikki Whiffin's group that just came out recently. Some of you might have seen it. And here they found that a single variant in a non-coding RNA was explaining 0.5% of all undiagnosed individuals with neurodevelopmental disorders in Genomics England. And to me, that is quite a mind-blowing figure to find a recurrent variant at this kind of sort of point in time after we've done so much sequencing. And this is why Professor Diana Brearley, my supervisor, is really sort of pushing all of these initiatives, particularly within the UK, to sequence RNA in large rare disease cohorts, both at the national level, within the 100,000 Genomes Project, as well as locally. And some of the earlier work from the lab, they started with RT-PCR. So in this case, 257 clinically suspicious variants of uncertain significance from across the UK. These were sent in by clinicians. And these were both coding and uncoding variants that were looked at. And like I said, most of them had RT-PCR. A subset had RNA sequencing. And basically, from this earlier study, we saw that about a third of these variants were affecting splicing. But most importantly, I think one of the biggest conclusions that came from this early work was that it's really hard to predict what the effect of these variants will be on the RNA sequencing until we actually look at the RNA sequencing. And so then from RT-PCR, we move on to RNA sequencing, some short-read sequencing. And this is one of our preprints that's under review at the moment. It's a subset of our splicing and disease cohort. And within this work, we were able to assess 71% of variants of uncertain significance that were sent to us. And we also identified 4 new potential diagnoses in patients that didn't have a candidate variant by detecting novel splicing events. However, what this data also showed is that we have some limitations with the short-read data. And what I found is that there were some events, so these were 3 of the variants that caused some events that I thought were consistently missed by the splicing tools. And this was for different reasons. So one of them was a low number of reads that were supporting the events, and this could be due to leaky splicing, NMD. We also had some intron retention that wasn't being picked up. And so then, of course, the next logical step was long-read sequencing. Can we use long-read sequencing to clarify some of these events that we weren't able to really clarify with the short-read or RT-PCR? So the samples that have been sequenced, they came from our splicing and disease cohort. For the most part, they had variants that we knew would cause aberrant splicing. For the most part, there's a couple of unknowns in there. And these had, again, there's some that are still getting RT-PCR, RNA-seq, but we wanted to have variants or samples that had RT-PCR, short-read RNA sequencing, and long-read sequencing. And most of our work really centers around using blood because it's the most easily accessible tissue. And so whole blood was collected in PacGene tubes, RNA was extracted, and then it was sent off to sequencing at PacBio. So I think it was the California and London lab. And so they did all of the library prep and sequencing. And so to date, we've had 2 successful runs. In our first run, we had 12 unique samples that were sequenced, and we had 4 pools. Each pool had 4 samples. And so for pool 1, 2, and 3, they all had globin depletion. And for pool 4, we had the same biosamples as pool 3, but no globin depletion. So this was done late last year. And then our newest batch that has just been sequenced, I think, a couple of weeks ago. In this case, we also had the 12 unique samples, but we didn't have the globin depletion, and instead we had 3 samples per pool. And once the samples were sequenced, they went through the standard isoseq pipeline, and then using Pigeon to filter the transcripts. And these are the results from our first batch, and Anna very kindly explained what each of the different categories meant in the previous talk. So we're mostly interested, or at least for now, at looking at those full splice match, the incomplete splice match, novel in catalog, and novel not in catalog. So for this first batch, on average, we've had 52,000 full splice match transcripts. And I've also colored in with the filtered and unfiltered just to see the effect that that Pigeon step has on the transcripts. And the same thing, but for our second batch, which did not have the globin depletion, we have a much higher diversity of full-length transcripts that we see, about 88,000 on average. And for us, it's really important that all of this is actually picking up genes that we are interested in looking at that are clinically relevant. And so what I did was that I just looked at the full or incomplete splice match for OMIM genes and panel app genes, which are the ones that are most commonly used within the diagnostic setting in the UK. And what I found was that on average, about 60% of OMIM genes were picked up and 63% of panel app genes for this first batch, which was the globin depleted batch. For the second batch, which did not have the globin depletion, there's an uplift here, and we've got, on average, 63% of OMIM genes that are picked up, and I think it's 67% of panel app genes. And this is comparable to what we get with the short reads. And this is, I only have this table for our first batch because we have all of the technologies now for it. And these are a list of the samples and the variants that we were looking at. And I've color-coded them. So the green were those cases for which PacBio actually gave us additional data. And we can see that's about half of them. The ones in black, we basically got the same results across all 3 technologies, but this is due to the effects of the variant. It's not necessarily due to sort of the biology or anything. So we could say it's probably the same or better. And then in the pink or red are the ones where the long reads didn't necessarily give us the same results. We didn't have enough coverage, or particularly for the COX7B and PHF8. That being said, in the second batch, I actually took a look at these genes, and I did find full-length transcripts for both COX7B and PHF8. So it was a matter of depth. The second batch had almost double the sequencing depth as the first one. So if this just had a higher depth, they'd probably either be all green or black. And I thought I'd show you some really interesting examples of what PacBio could, or what this data could tell us. So this is a case that had a variant in this RPS7 gene. And the RT-PCR results came back normal for this particular case. And within the UK, RT-PCR is what's done in the clinical lab. So this would have been reported back to the clinicians as a normal sample. Hopefully you can maybe see it. This is a sashimi plot for those of you that might not be familiar. The sort of peaks are the coverage of the exons, and the lines across is showing you how many reads are covering those junctions, and so how much support we have for those particular junctions. And so here in the arrow, you see that We see some potential intron retention with the Illumina short reads, but we can't clarify this just with the short reads or with the RT-PCR because, as we saw, that came back normal. And additionally, something else that I noticed with the short reads is that we've got a lot higher number of reads covering this particular gene. We've got over 1,000 reads covering the junctions, whereas for the normals or for our controls, we've got about 100. And so this is what the data with PacBio looks like. See if I can get my little pointer again. So we've got the controls, 2 controls at the top, and hopefully the people in the back can see the proband. We've got the variant right here. It's a +3 variant. And effectively, what we can get from this data is that there is intron retention, or the variant is found in all of the transcripts that have this intron retention. However, that's also the case for the controls. They also have a transcript with intron retention, and this is probably why the RT-PCR came back normal. However, fortunately for me, this was a transcript, or this was a gene that only had 2 transcripts, so I could go manually and back into the data to look at this in particular. And I compared it to the rest of the samples that were in its pool. And so this particular gene had 2 transcripts. One that had that intron retention, and the second one was just the main select transcript. And on average, it was a 50-50 split between this intron retained transcript and the main annotated transcript. However, for our particular case, the intron retained transcript was almost 40 times as high as the main annotated transcript. So this is kind of giving us a bit of insight on the biology of maybe why this is causing this particular disorder. This other case, we've got a homozygous deletion, 22 base pair deletions within this KLH-L7 gene. And here are all the transcripts that were identified with PacBio for this gene. What I found interesting was that we had a full splice match to the reference, and to me this is odd because this is a homozygous deletion, so I would have expected not to see any full-length transcripts. But we also saw that, so that's pointing to where the variant is, and we also saw the transcript that we were seeing in the short read, which is the exon skipping of that particular exon again. So this is the exon that was affected, and so we don't see that exon in this transcript, and that's what we saw with the short reads. I took a bit of a closer look in IGV just to see why were we getting this normal transcript. And first off, what I noticed is that there seems to be a bit of mismapping for this particular case. In blue is where that deletion falls, and that's really what the read should look like. But I think if you notice that we've got this GGT here, and we've also got a GGT there, so that's just probably a mapping error. But what that also tells me is that the canonical donor site is still present. And so this could be some potential leaky splicing that I don't think we would have ever even thought to kind of interpret with the short read data. And then the last example I'm going to show is for this particular gene, SF3B4. We just had a variant that was in this gene. And with the Illumina data set, we're only able to really look well the first 2 exons. The rest just weren't covered well. It's a high GC-rich gene. And this is what that particular gene, that's what the transcript looked like with the PacBio data. So really nice and clean, really high depth. So already just a huge advantage. And so just to sort of summarize what I've kind of wanted to talk about here, some technical aspects. The Kinnex kit was actually able to pick up at least 60% of OMM genes and 63% of panel-lab genes in blood. And I was very conservative with this because these are a full splice match and incomplete splice match that doesn't include the novel in catalog or not in catalog. This might be controversial because sometimes I think we just assume that we have to have globin depletion in blood samples, but actually having a higher sequencing depth and no globin depletion was, I found, a pretty good option. And this is really, really key. The file sizes were reasonable, and it's comparable to the short reads. And I say that this is key because as someone who's trying to integrate short read RNA sequencing within our health system, I think Data storage and data processing is something that people don't really think about when they start the project. We're just going to sequence a bunch, but having somewhere to store the data, having someone to process it, that costs money. And currently, the systems in place just aren't prepared to deal with huge amounts of data, not even to store or to process it. And before I talk about the benefits, of course, this is going to have the same limitations as looking at blood as a tissue in short-read sequencing. But in this case, the benefits of using long-read RNA-seq is that we're able to detect novel transcripts. I think it's been quite clear that we always see way more than we expect. And for me in particular is the intron retention. This keeps popping up, and it's just something that we just don't pick up really well with the short read. We've got better resolution of complex regions like the SF3B4 gene. And we're able to quantify transcript abundance. And this is going to be really useful for us to look at molecular mechanisms, really start to look at nonsense-mediated decay and leaking splicing, and really start to kind of think about this in terms of the therapeutics. Something I haven't done yet is the phasing of allelic variants. I think it'll be much easier to look at monoallelic expression, particularly for the undiagnosed cases that we might want to start picking up different things within the data. And more importantly, if we know where splicing is going wrong, we can start to develop therapies to treat these patients. So our next steps, we have some patients that are still undiagnosed, and so trying to sort of adapt our short read methods into the long read, for the long read data is one of those steps, and then hopefully using this to identify new diagnostic candidates for these patients. And then just sort of overall, identifying what is best to employ long read versus short read versus RT-PCR, because like I said, there's still some limitations. What tissue, what are we able to pick up, what exactly it is that we're looking for. And then lastly, I'd just like to thank everyone that has been involved in the project, particularly my supervisor Diana and all of the PacBio team that has really helped us get through this and look at all of the data as quickly as we've been able to.